Thank you to the organizer to give me this opportunity to present my work in 10 minutes. So yesterday we have seen like how Dr. Sheshadri so clearly made it clear that it's very important that we aim to prevent dementia rather than curing it, and the importance of studying healthy individuals to understand more about the treatment for dementia. So I would like to introduce to Mr. Imaginary. So Mr. Imaginary is 72 years of age, he's father of 3 daughters. Recently has been diagnosed with Alzheimer's disease, and he cannot recognize his daughters anymore, unfortunately. And the prescribed medicine so far hasn't worked well in his to improve his condition. Now, the condition, like similar to Mr. Imaginary, so is on the rise. So in every 4 seconds, we have a new case of people suffering from dementia. Now, in one point, we would all agree upon the treatment in case of Mr. Imaginary didn't work well because simply he was diagnosed at a stage where he developed already a strong cognitive impairment. But he underwent through a phase where he was at risk of dementia, and if the proper therapeutic interventions could be applied at this stage, maybe we could reverse his condition and we could impede his transformation to develop this strong cognitive impairment. So that's why we aim for biomarker findings. But for that, we need to understand what also happens in the molecular level. Now, we know from the other studies that when we see the dementia patient and we compare them to the non-dependent control, we see the molecular changes already happens at when the disease is onsetting. But these changes also starts happening years before the onset of the disease. As you can see, around 20 years or so before the symptoms appearing. So the molecular changes is there. So we are still missing the right biomarker to find individuals who are at risk of developing dementia. So we aim for finding that to stratify subjects at risk of developing dementia at late age. So we thought like total blood would be a good source of biomarker because it's very easily accessible. And in this study, we will focus on microRNA as a potential biomarker. Now, why microRNA? It comes solely because of its mode of action. We know that it's synthesized in the nucleus, and then it's processed there, and before it's transported to the cytoplasm. So where it's even further processed, and then it's carried, and it can then be transported by an enzyme called AGO2, a protein, and then to the cognate mRNA it binds, and then can either degrade or repress the transcription, leading to the translational repression. So that means that the microRNA binding to the cognate mRNA would lead to the translational inhibition of that corresponding mRNA. Now, the good thing about this microRNA as biomarker is that they are very stable in different conditions, and they can interact with many partners. I would like to emphasize on that by giving this example where we modulated one single microRNA. We inhibited this microRNA in the prefrontal cortex of mouse brain, healthy mouse brain, and we have seen that number of processes, different processes are being actually affected. And as you know, dementia is a complex disorder getting affected by different biological processes. So microRNA, as you can see, that can regulate these different processes are definitely of great importance to look into. Also, more on this you can find in poster 21. But we have seen that these mice also develop schizophrenic symptoms. So as you can imagine, like, so how it's important to analyze microRNA. Now, in this study, we have two folds of aim. So first, we want to identify individuals who have mild cognitive impairments or at risk of developing dementia. And second, if we can treat these individuals to restore their memory. So here, like, we want to learn from the healthy individuals. So what we know that some of the cognitive features already starts declining at early time points, at early 30s. So we had the access to a project called Psychos, where we are collecting human, we are recruiting them, and then we collect their blood, and we also have information about their cognitive performance. So what we could ask that we could take their blood microRNA expression in these healthy individuals who are aged between 21 to 30 years of age. So as you know, so they are still cognitively healthy, but there is some inter-individual variability. Our question is if we can explain with microRNA this inter-individual variability in these individuals. So what we are looking at, the expression of microRNAs, and we let the microRNAs to form different clusters solely based on the expression of the microRNAs. So the microRNAs which are highly correlated, they belong to one cluster, and this cluster of microRNAs is not that correlated to the other cluster. And when we define this cluster of microRNAs, then we can ask that how this microRNA cluster is correlated with cognition. Now what we see, basically, we could find three major clusters significantly linked to cognition, two being negatively correlated, the other is positively correlated. So we see basically possibly a causative and compensatory response mechanism, like in terms of microRNA. We wanted to look further into that in terms of biological process by taking the brain-enriched microRNAs, and when we look at the biological process, we see aging is coming as a major biological process in these individuals. So that means that although these individuals are young, they are asymptomatic, they do not develop any symptoms of cognitive decline, but we see aging response in microRNAs are there to explain their inter-individual variability. So we wanted to study aging into further extent. So to do that, so we looked into a longitudinal setting now in a mouse. So we followed mice like in terms of cognition from 12 months to 16.5 months, and we also collected blood from them in every 6 weeks. What we have seen that the mild cognitive impairment and first appear at 16 months of age, where mice is taking more time to find out the hidden platform in water mesh settings. Now, we also sequenced the blood small RNA from this mouse study, and we could ask if we can run some machine learning model, like to get the features from the data. So we want to learn from the data, and then we want to see if this microRNA features is also conserved in our microRNA clusters, which we found from the asymptomatic human data. So therefore we actually found some conserved cluster of microRNA consisting of 3 microRNAs, and we want to test this 3 microRNA based cluster into disease condition and also at risk conditions, and compare it to the healthy subjects. Now first we want to look at in the mouse longitudinal setting. So as we have seen that these mice develop cognitive decline significantly at 16.5 months of age, but interestingly we see an increased expression of our cluster already at 15 months, where the cognitive impairment was still non-significant. Now we had this also I would like to point out that this was a longitudinal study, but we also had this Psychos project where we could address similar question but to the cross-sectional subgroups of human subjects. So what we have seen that in the age math, like comparable age groups, so we have seen there is a decline in terms of weighted cognitive score, or at middle age, like it's declining, but it's only getting significant at p-value 0.07. But in terms of microRNA expression, we see already at this middle age, this cluster of microRNA increased significantly. Now what do we learn from this? We learn from this, we see a consistent pattern in mouse and in human, that a cluster of microRNAs is increased prior to cognitive decline. Now we wanted to ask if this increased expression we would also see in disease conditions. So first we are looking at now mouse in an APPS1 mice, like it's a model of AD, and we see that in the transgenic mice the expression of this microRNA is increased at 4 months when the cognitive impairment is still mild. And at 8 months, when the cognitive impairment is dramatic, so we see still increased expression. In human, we see in MCI patient in plasma, the expression of the same cluster is increased, and also in CSF, which is the proxy for the brain. In this case, in MCI patient, the expression is still increased. So we see, therefore we find a cluster of microRNAs now, which is able to detect the individuals at risk of developing dementia, and also we see them the same pattern in disease conditions. Now the second aim of the project, so we wanted to rescue the individuals who are at early cognitive deficits. Now we wanted to look for these therapeutic interventions by taking microRNA from these three microRNA-based clusters based on its expression in neuron and its role in synaptic function. Now we focus on this microRNA, which is very highly enriched in neuron, and when we manipulate it, we overexpress this microRNA in a primary culture dish of hippocampal neurons. We see the downregulated cluster coming from these mini-treated ones are regulating learning and memory processes. So it is very interesting. We thought so maybe this overexpression is causative to the cognitive decline. Now, since we are mostly interested for the treatment, so what we asked then, if we could rescue or restore memory in a memory-impaired mice. So we took 3 months and 16 months old mice. When we normally compare this young and old mice, we see a memory impairment at 16 months. But when we now inhibit this expression of microRNA, so we see we could restore memory in this old mice. So the old mice becomes juvenile in terms of cognitive performance. So I'd like to summarize with that we find in our study 3 microRNA-based clusters, so which are informative about cognitive status, and we could restore memory in a memory-impaired mice using this microRNA. The thing we do not still know that what's the source of this microRNA in blood, if it's coming from the brain or from the peripheral system. But yeah, so that's the place where future studies can address. And with that, I'd like to conclude and thank all the authors of and the contributors of the work, and especially I would like to thank Professor Schulz, Falkai, Professor Sanan Benesi, Professor Andre Fischer for their excellent support for this project. So, and I'd like to thank you, to be here to listen to this talk. Thank you.
Thank you very much. Questions?
Very nice talk, thank you very much. I did not really understand how you identified the 3 microRNAs. What was the analytical and mathematical approach?
Yeah, so basically, so for this, so we applied, we took help from both human data and also from mouse data. So as I have shown that, so from human data, we ran an analysis called gene co-expression analysis. So basically, we wanted to know which clusters are linked to cognition. And then from mouse, so we to get the signature, so we ran a machine learning feature selection method. We used two independent methods for selecting features from our mouse data. And then so we overlap, so what is the overlap between the feature that we are getting from mouse and what's the overlap with the microRNA cluster that we have seen that is being linked to cognition. And later, I haven't addressed that, but later to even to make it more narrower to 3 microRNA-based signals, so we also compared if the targets of these microRNAs are also conserved in GWAS studies where they have linked certain genes to be involved in general cognitive function. So we see the targets of these 3 genes are significantly overlapping with the genes that have been described as a risk factors for cognitive function. So we ran several measures to determine these 3 microRNAs, and we also know that these 3 microRNAs are equally expressed in both brain and blood. Yeah, thanks.
Very interesting talk. I've got a question. If you inject this microRNA, do you also see changes in the general appearance of these animals? So might this also be something more general associated to age, not only cognition? So
general what?
Do these animals also change in their general appearance? Are they more healthy than their littermates after you injected the microRNA first?
Yeah, that's a very good question. I'm like, so in this case, like we haven't like measured other features. So we are particularly interested about their cognition. But we see when we sequence them, so we see a major change in terms of many things, like in affecting also like extracellular matrix and other things. But we haven't like phenotyped them into great detail, like to know how that is also affecting in overall phenotype. So here, as a phenotype, we only looked at in their cognition.
And have you checked whether the expression of this microRNA changes if you use interventions which improve cognition, such as enriched environment?
Yeah, that's a very good question. So here we are focusing on 3 microRNAs. Not all of them change, so we also looked into that. So for example, we also study effect of exercise in cognition. So we see like 1 microRNA from this cluster is also changing there. And what I haven't addressed that like this microRNA cluster is also conserved. We see their targets are conserved not only in AD but also in FTLD, in Parkinson's disease. We have seen also in the proteomics of AD. So we believe that like so this cluster is more like a general dementia. Like it's not specific to AD, but more to the general cognitive dysfunction.
Thank you.
Okay, I think because of also the time we should move on. Thank you very much for this excellent presentation.